Hello class! Today we will be learning about auxin and its use in tropisms. So here I'm drawing the plant's outer cells, and we call these cells the coleoptile cells, and here are the auxin. The auxin. So what exactly is auxin? Well, auxin is the group of any chemical substance that promotes coleoptile, that's right, plant cell elongation. Okay, so the most common auxin is actually IAA, which is indoleacetic acid, and auxin's actually a plant hormone. Yay, guys! You know, hormones. What fun!
On that happy note, auxin is actually produced in the embryo of seeds and in young leaves and in the meristem of apical buds, which are the primary growing parts at the tip of the stems of the plant.
So you guys might be wondering, what exactly is the pathway through which auxin influences all these tropisms? Well guys, let's see. Auxin actually increases the activity of proton pumps in the cell membranes of plant cells. So through active transport, this proton pump pumps a bunch of protons out into the cell wall. And because there's a huge concentration of protons now in the cell wall becomes more acidic. This acidification results in the activation of wedge-shaped expansins, which are basically proteins. They separate cellulose microfibrils in the cell wall from cross-linking polysaccharides. And basically that just results in the cell wall being more flexible and being able to elongate itself. And in addition to that, the membrane potential is increased, so ion uptake into the cell is also increased, which results through osmosis for water to flow into the plant cell and expand it. So if we want to go more in depth, you can remember that auxin is actually a plant hormone, so that means it's part of the endocrine system. So auxin binds to its specific auxin receptor and activates it, and this activated complex binds to a degradation complex, and this entire giant thing manages to activate a gene regulator that is already binded to an inhibitory protein, making the inhibitory protein degrade and leaving the gene regulator by itself. So this activated gene regulator is able to go inside the nucleus and bind to DNA to regulate transcription and translation of specific proteins that auxin controls.
Okay, so you might be wondering how exactly auxin plays a role in tropisms. And so one of the ones that auxin plays an important role in is actually phototropism, which is the attraction of like a plant towards or away from a light. And towards light would be positive phototropism, whereas away would be negative phototropism. So here we have a plant stem with little auxin hormones in it, these little pink things you see right here. And basically what happens is that auxin is evenly dispersed until you have sunlight. And once you have sunlight, auxin goes to the shady side of the plant, which from previous reference, you know that when auxin moves to the shady side, it will cause cell elongation. so now you see you have these plant cells in this stem with auxin inside these plant cells, and through the process described earlier, this promotes the cell elongation of the shady side cells, which again are coleoptile cells. And here you just see how this increased turgor pressure due to water potential elongates these cells, make them more flexible, and as such, this plant starts tilting towards the light. So you can see here that plants exhibit positive phototropism. Okay, so from here we get a couple of really important experiments, such as the Darwin experiment and the Boysen-Jensen experiment, which is kind of an extension of the Darwin experiment. In the Darwin experiment, basically what he was testing was how auxin played a role in this leaning towards or away, or positive or negative phototropism. And he was testing whether or not a seedling would lean towards this light source or away from this light source if it had this tip of the coleoptile, which again, the coleoptile is a sheath that encloses the shoot of a sprouting grass seedling. So as you can see in the picture is that a normal seedling would have positive phototropism and lean towards this light source, whereas this tip of the coleoptile was cut off, it would not lean towards it at all. And the same goes for if this tip of the coleoptile was covered with an opaque cap, or light did not come through to the tip of the coleoptile, then the seedling would not lean towards the light source either. So, but if it had a clear cap and light was allowed to pass through, it would still exhibit positive phototropism. So basically what this led him to conclude was that this tip was responsible for sensing light, but the growth response that occurred for positive phototropism actually occurred a distance away, supporting an idea that this was a signal transduction pathway.
Boysen-Jensen supported Darwin's experiment by performing the same experiment, except he used a block of gelatin, which is permeable, and an impermeable block, microblock. And this showed that the plant response was triggered by light-activated chemicals.
So now we have another tropism. In this case, it is thigmotropism. And thigmotropism is basically the movement in which a plant moves or grows in response to a touch or a touch stimulus. And these are transmitted through action potentials in plants, which are kind of like nerve potentials in animals with calcium. And they are transferred through plasmodesmata, the gaps between two plant cells. So, in the case of, let's say, the plant's vines, they also use auxin as well. And basically, like with phototropism, the side away from the stimulus is where auxin congregates, elongating the far side. And the mimosa plant also uses thigmotropism. When touched, these pulvini or motor cells either lose turgor pressure or gain turgor, depending on their position. These motor cells either lose or gain potassium, again causing water to leave or come into the cell respectively by osmosis. Evolutionary, vines that exhibited thigmotropism were able to grow taller and stronger with the help of the support, and resulted in the plant receiving more sunlight. And the same goes for the mimosa plant. Maybe strong winds stimulated the plant to close up its leaves to conserve water necessary. And both of these adaptations in plants allowed them to survive and reproduce more often.
Gravitropism is the response of a plant to gravity. The plants detect gravity through stadoliths, which are actually plastids containing starch grains. And these grains triggers calcium ions and auxin to accumulate towards the tips of the roots. And as a result, the roots display positive gravitropism, and the shoots will face upwards and display negative gravitropism. And the starch grains and statoliths fall onto the upper half, the sides, and the lower half of the cell. However, only the lower half of the plant cell membrane is responsive, therefore causing the roots to grow downwards.
So, we hope that's helpful. Emocon.